Good day, everyone. So we're reviewing Wetzel's Limnology textbook. This is edition number 4. We're on chapter 15, the phosphorus cycle, and more importantly right now, we are looking at the amount of phosphorus needed for growth. And this was the chapter we left it at. Phosphorus concentration required for growth. Of course, this recycling and utilization of stored phosphorus reserves cannot persist for very long. Losses continually occur from the colloidal phosphorus component, as well as from sedimentation from the particulate phosphorus component, and with outflows of both dissolved and particulate components. Inputs of phosphorus to the system are needed continually either from the littoral zone and turbulent transfer from lower depth strata and sediments or externally in order to sustain growth for extended periods. The amount of phosphorus input or loading then is important to the sustenance of algal growth and is one of the many factors influencing the types of phytoplankton that are growing in a particular lake at a particular time of year. It is therefore more relevant to the question of increasing phytoplankton productivity to view phosphorus concentration in terms of total phosphorus, since most of the phosphorus is bound in the particulate component at any given time. From the few studies available in which common phytoplankton species of lakes of different productivity were studied in relation to phosphorus requirements, the minimum phosphorus required per cell volume can be evaluated. The colonial diatom Astarionella formosa is often found in oligotrophic waters as a low phosphorus requirement. It can reach maximum cell densities at very low phosphorus concentrations. The quantity of phytoplankton expressed as cell volume that may be produced by 1 microgram of phosphorus per liter is between 2 and 5 millimeter cube per liter. This quantity of algae is common in lakes of low productivity. Tabellaria and Fragillaria are diatoms that reach maximum population densities at phosphorus concentrations of approximately 45 microgram of phosphate as phosphorus per liter, while Cynidismus approximately needs higher concentration of around 500 micrograms per liter. The Cyanobacterium oscillatoria rubescens does not reach its maximum phosphorus content until the initial phosphate concentration of the media used is about 3,000 micrograms of phosphate as phosphorus per liter. It is of ecological interest to note that cations stimulates phosphates uptake by the Cyanobacterium Cyanococcus, which is equal to Anacystis. Calcium ion in particular cause a pronounced reduction in the half saturation concentration for orthophosphate uptake by increasing the active transport of phosphorus into the cells. Before we move on, let's look at table 15-12, the minimal phosphorus requirements per unit cell volume of several algae or cyanobacteria common to lakes of progressively increasing productivity. So we have algae on the left, and then we have the minimum phosphorus requirement in milligrams per millimeter cube of cell volume. So on the low end, we have Astarionella, as we just saw in the text, and on the high end, we have Tabellaria, which is like I believe it's a diatom. And then in this table we have both algae and cyanobacteria. Anyway, Microcystis, I believe, is a cyanobacteria, but a lot of them require a fair amount of phosphorus, like Cynidismus, Oscillatoria, Microcystis. They require more than 0.5 or 0.5 millimeter cube. Studies on the kinetics of phosphate uptake and growth of Cynidismus in continuous culture have shown that phosphorus uptake velocity is a function of both the concentrations of internal cellular phosphorus compounds and the concentration of the external substrate. The apparent half-saturation constant, Km, the coefficient of the Michaelis-Menten formula was 0.6 micromole per liter, approximately 18 micrograms of phosphorus per liter, for phosphorus uptake, whereas the apparent half-saturation constant for growth rate, the Monod equation, was less than Km for uptake by an order of magnitude, Growth was a function of cellular phosphorus concentration. Internal polyphosphate content appeared to regulate growth rate, particularly in the initiation of cell division. The activity of alkaline phosphatase, the primary enzyme involved in the release of phosphates from polyphosphate, exhibited a relationship that was inversely related to growth rate and was correlated to both polyphosphates and internally stored surplus phosphorus. During phosphate limitation, polyphosphates are mobilized for the synthesis of cellular macromolecules. Similar results were found for species of diatoms and for green algae and cyanobacteria. Phosphorus uptake and competition by phytoplankton versus bacteria. Early studies on the uptake kinetics and competition for phosphorus between phytoplankton and bacteria were inconclusive. For example, bacteria were found to have a lower affinity, Vm over Km, based on cell surface area, for phosphate than did two diatoms. This implies that diatoms can transport more phosphate per unit of cell membrane than bacteria at low phosphorus under the experimental conditions. However, bacteria could potentially outgrow the diatoms in situ because of a more favorable surface area to volume ratio, and indeed, bacteria had higher growth rates in single species culture under the experimental conditions. As cell size increases, volume increases faster than surface area, so the amount of phosphorus applied from a unit of the outer membrane to a unit of cell volume diminishes. To better reflect competitive abilities, the affinities in table 15-13 can be expressed per unit cell volume by multiplying values by the cell's surface area divided by the volume ratio, which would be 3 over R for a sphere and 2 by L plus R over L over R over LR for a cylinder where radius is R and L is length. Assuming the bacteria in table 15-13 are cylindrical with radius of 0.5 and length of 1 micrometers, and the diatoms are spherical with radius of 10 micrometers, their mean affinities of 1.23 and 3.8 by 10 to the minus 6 liter per micrometer square per day would become 7.4 and 1.1 by 10 to the minus 6, with bacteria now having higher transport affinities based on cell volume. Morlott and Brown found that chlorophytes and a diatom had lower volume-based phosphate transport affinities than several small and large prokaryotic cyanobacteria, and a yeast suggesting that the algae were not as competitive at low phosphorus concentration. These studies suggest that if competition between pelagic microbial heterotrophs and eukaryotic algae were based only on phosphates, then the former would dominate and eukaryotic algae would be excluded. However, microbial heterotroph abundance in situ depends on continual supplies of organic carbon, and thus the abundance may be limited to some extent by organic carbon supplied by phytoplankton. Intensive grazing of the smaller bacteria and picocyanobacteria by microzooplankton may also allow larger eukaryotic algae to thrive in low phosphorus environments. A plethora of studies have examined the phosphorus uptake kinetics and specific growth rates of bacterioplankton, which includes photosynthetic picocyanobacteria and picoeukaryotic algae, in comparison to those of larger phytoplankton. Correlation analysis and direct measurements of specific growth rates in relation to phosphorus availability and competition among bacteria and phytoplankton indicated these primary points. A. Bacterioplankton have substantially higher phosphorus requirements than do larger phytoplankton. B. Bacterioplankton net consumption of phosphorus is larger than that of the phytoplankton. C. Bacterioplankton contains much greater amount of phosphorus than algae. D. Phosphorus as well as, or rather than, organic carbon commonly limit bacterioplankton. And E. Bacterioplankton can outcompete algae for phosphorus under a wide range of phosphorus supply rates. Nevertheless, since these two groups coexist, some factors other than phosphorus constrain heterotrophic bacterioplankton populations in situ. Since the bacterioplankton community is operationally defined by size fraction and will include picosaccharobacteria and picoeukaryotic algae, which are not heterotrophic, the extent to which organic carbon limits heterotrophic bacterioplankton populations remains unclear. Phosphate is the preferred form of phosphorus for uptake into both phytoplankton and bacteria. Phytoplankton use both phosphate and dissolved organic phosphorus compounds, particularly at high substrate concentration. At most natural concentrations, however, the uptake of phosphate is completely dominated by bacteria. The phosphorus contained in the bacteria is then regenerated by death and autolysis of the cells or by feeding activities of heterotrophic flagellates. Because bacteria have high requirements for phosphorus, much of the phosphorus occurs in and is regenerated from these phosphorus-rich particles. Table 15-13, phosphorus-dependent growth rate and phosphorus uptake kinetics of 2 diatoms and 3 bacterial species. So the diatoms are on the left here, Cyclotella and Thalassocerea fluviatilis, and this is the Cyclotella nana. So we have mean cell volume, mean cell surface, AO, which is the minimum phosphorus content per cell, micro M, which is the growth rate during unrestricted growth at 22 degrees centigrade, Km, which is the Michaelis half saturation constants or of uptake mechanism. Then we have Vm, which is the maximum uptake rate for orthophosphate phosphorus per unit area of cell surface. And then so we have Vm over Km, which would mean the slope of Michaelis-Menten uptake curve near the origin, which represents relative competitive ability to sequester phosphorus at low concentration. So the ability to sequester phosphorus at low concentration. So I'm assuming when that ratio is high, that means that that the cells are able, or the bacteria or the diatoms are able to sequester phosphorus at those high concentrations. At those low concentrations. Phosphorus and eutrophication. Eutrophication and trophic status. All phytoplankton species require phosphorus, nitrogen, major ions, several trace metals, and in some species, silica. The importance of phosphorus to lake ecology lies in the fact that in most unpolluted lakes globally, phosphorus is in shortest supply. Therefore, an increase in phosphorus inputs results in higher productivity, a phenomenon referred to as Liebig's law of the minimum, or the principle of limiting factor. Today, it is sometimes referred to as the phosphorus paradigm. The majority of monitored oligotrophic lakes globally are phosphorus limited, although some are not. For example, the nitrogen limited lakes in low nitrogen deposition areas in northern Sweden and lakes in the arid American Great Basins. Some oligotrophic lakes may be limited by iron or co-limited by iron and phosphorus, or at least some of their communities with high metal requirements such as picocyanobacteria may be growth limited for short periods of time. In naturally phosphorus limited lakes that have become nutrient enriched or eutrophic, phosphorus is usually the only nutrient with natural or background levels low enough for it to be the basis of effective nutrient control programs that lower anthropogenic inputs to restore or maintain oligotrophic to mesotrophic conditions. For this reason, phosphorus has become the cornerstone of eutrophication management. As whole lake phosphorus fertilization experiments in the experimental lakes areas have repeatedly shown, even dilute, low solute lakes have natural major ion and trace metal concentration high enough to support large increases in productivity. Nitrogen is a special case because should a eutrophic lake become nitrogen limited, diazotrophic nitrogen fixing cyanobacteria will convert dissolved nitrogen to ammonia during warm periods in the summer, provided that trace metal cofactors are high enough to support heterocyst and nitrogenous synthesis. Figures 15-12. photograph of Lake 226 of the experimental lakes ELA of northwestern Ontario, which was partitioned at the construction of the basin on 4 September 1973. And David Schindler, director of ELA from 1968 to 1989, installing the curtain, write, "The far northeastern basin, fertilized with phosphorus, nitrogen, and carbon, was covered by a dense algal bloom within 2 months. No increase in algae or species change were observed in the near basin, which received similar quantities of nitrogen and carbon, but no phosphorus." So this is with nitrogen and phosphorus, this is with just nitrogen. And you can see that we have a bloom in the area where we have where we have nitrogen and phosphorus, and we don't where phosphorus is missing or has not been seeded inside that portion of the lake. And then this is the boom that they use to prevent the water from the phosphorus and nitrogen seeded part of the lake to move to the place where to the part of the lake where phosphorus was absent and was not present in the seeding mechanism, at least probably present naturally to some degree, although probably very small concentrations. As phosphorus inputs increase, lake characteristics progressively change, with oligotrophy signifying a state associated with low nutrient concentrations and eutrophy signifying a state associated with excessive nutrient concentrations. Mesotrophy is in the middle of this continuum. Naumann introduced the general concept of oligotrophy and eutrophy and distinguished them on the basis of phytoplanktonic population, a recognition that as lakes become nutrient enriched, they become more productive. He later elaborated on the concept. Naumann emphasized that chemical factors, particularly phosphorus, combined nitrogen, for example, ammonia, nitrite and nitrate, and calcium were primary determining factors of productivity rates, although today it is recognized that eutrophic saltwater lakes with low calcium are similar in production and other characteristics to eutrophic hardwater lakes. Limnological evidence since Naumann's early work has shown a strong relationship between the biological dynamics and structure of a lake and its concentrations of nutrients. The consequences of eutrophication, that is the development of characteristics that differ markedly from oligotrophic lakes, such as loss of macrophyte beds, hypolimnetic oxygen loss, dense phytoplankton population, with the formation of harmful cyanobacteria blooms, and changes in invertebrates and fish community structure, have been further elaborated on by many others, including Thiemann. The terms oligotrophy for lakes of low productions and eutrophy for lakes of high productions evolved from similar concepts developed for bogs. So before we move on, let's look at this picture here, figure 15-13. So photograph of nitrogen fixing cyanobacteria bloom in lakes 303 in the experimental lakes area in northwestern Ontario on 1 September 2019, 12 weeks after the first weekly phosphorus addition. Although the lake was not fertilized with nitrogen during the experiment, nitrogen fixation doubled the total nitrogen concentration during the bloom. So here we have a cyanobacterial bloom that has happened after the lake was seeded with phosphorus, but not with nitrogen. So therefore, the point is that the cyanobacteria fixed nitrogen, meaning by that they immobilize or they synthesize nitrogen from nitrate and nitrite probably from atmospheric nitrogen, and helped the bloom to develop and flourish. Today, the trophic state concept is based on the notion that nutrient enrichment and production biomass are causally linked and does not try to incorporate all of the biological and abiotic consequences of that production. The concept has led to a useful system for classifying the extent of nutrient enrichment of a lake based on phosphorus concentration, with 3 and sometimes more categories used: oligotrophic, mesotrophic, eutrophic, and hypereutrophic. The phosphorus boundaries separating them are somewhat fluid, being based on lake characteristics that change with enrichment. In addition, different jurisdictions use different boundaries. While phosphorus and productivity run along a continuum, such classifications are very useful in public communication and environmental management. So that's going to be it for today. And tomorrow we'll pick it up with the combination of many physical and biological variables. Thank you for tuning in today, and I'll talk to you tomorrow. Bye.